Segment 3. Cone opponent cells in the retina and LGN. The lateral geniculate nucleus is a structure in the thalamus, part of the midbrain that receives input from the retinal ganglion cells and has input and output connections to the visual cortex. Many ganglion cells in the retina and the LGN of the thalamus are maximally stimulated by spots of light. Some of these retinal and LGN ganglion cells are excited by the L cone onset in their center and inhibited by M cone onsets in their surround. These L-M cells are one type of cone opponent cell, so named because different sources of chromatic information are pitted against one another. Cone opponent cell is a cell type found in the retinal, lateral geniculate nucleus, and visual cortex that, in effect, subtracts one type of cone input from another. Coniocellular, referring to cells in the coniocellular layer of the lateral geniculate nucleus of the thalamus. Conio from the Greek dus, referring to the appearance of the cells. S-cone signals go through the coniocellular layer. Parvocellular, referring to cells in the parvocellular layers of the lateral geniculate nucleus of the thalamus. Parvo from the Greek for small, referring to the size of the cell. M and L cone opponent signals are mostly found in the parvocellular layers. A different ganglion cell helps to keep track of day and night, the primary force adjusting your internal circadian clock in sunlight. Circadian, referring to cells in the magnocellular layers of the lateral geniculate nucleus of the thalamus. Magno from the Greek for large, referring to the size of the cells. We have learned that there is previously unknown photosensitive cells in the retina. It receives input from rods in the cones, but it also contains its own photopigments, so it can detect light even when normal photoreceptors are absent. Melanopsin, a photopigment found in a class of photoreceptive retinal ganglion cells. Its outputs go to sensors in the brain that control functions like your circadian clock. Step 3. Color appearance. Rods make a small important contribution of the color vision, but only in fairly dim light. The retina and LGN contain cells that have repackaged that information into cone opponent cell different signals that can train our ability to see difference between regions. 3. Number many colors. We have exactly 3 different types of cone photoreceptors. The light reaching any part of the retina will be translated into 3 responses, one for each local population of cones. It has been estimated that with the system we can discriminate the surfaces of more than 2 million different color space. The three-dimensional space established because color perception is based on the outputs of 3 cone types that describe the onset of colors. The three-dimensional space is the same as the height, length, and width of a cube. The limits of the rainbow. When we wrap the wavelength spectrum into a color circle, we join the red and blue ends with a set of colors that are called non-spectral hues, hues that can arise only from mixtures of wavelengths. Colors that are not included in the spectrums, all the colors of the rainbow. Brown is one of these colors. There are no brown lights. Brown is seen when a mixture of wavelengths that would look yellow, greenish yellow, and orange is seen in the company of the other, brighter patches of color. Opponent colors. Young and Helmholtz were describing a trichromatic theory with 3 basic colors, red, green, and blue. The opponent color theory is a theory that perception of color is based on the output of 3 major mechanisms, each of them resulting from an opponency between two colors, red, green, blue, yellow, and black, white. Gray can be described as blackish white. Unique hue. Any of 4 colors that can be described with only a single color term, red, yellow, green, blue. Other colors such as purple or orange can be described as compounds. The L-M cells aren't in quite the right place either. The L cone end of the axis is near perceptual red, but the M cone end is bluish green. Kraskov, Williams, and Healy call these endpoints the cardinal directions in color space, but they are not perceptual red, green, yellow. We need another transformation of color signals in order to make the cone opponent signals actually turn into color opponent signals.